All right, so today what we will go over is going to be general parts of the cell. We will go over some diseases of those parts of the cell, especially the lysosome, a little bit of mitochondria, a little bit of peroxisome, which is different than the lysosome, and then a little bit of, there's one disease for the cytoskeleton of the cell. And then after that, we will talk about the surface of the cell, how it has different receptors. There are three major families of receptors. Those are ion channel or ion gating receptors, G protein receptors, and the kinase receptors. And so they, in one form or another, will talk to a kinase. And there are three types of those kinase receptors. So that's what we'll be going over today. So starting with the general cell organelles. So kind of like organs of the body, these are organs of the cell. Organelles is what we call them. All right, so first, the nucleus. Nucleus is kind of a, it's almost like sticking a cell inside of another cell. It's its own little ball, and it protects the genetic material. So the DNA is in there. That way it's not going to come across any enzymes that might accidentally get loose and damage it, or bumping into other things, or toxins have another layer to go through. And so it's just a protective shell, and it also helps with regulation on when certain things will talk to the DNA. All right, so that's the nucleus, and from DNA we often get RNA, as we've talked about earlier, and mRNA is what we use to code proteins. And I'm going to go ahead and follow that story, since that's one that's kind of important to embed in our minds, to talk about the other organelles. So mRNA would be made in the nucleus, and then it's going to come out, and it's going to find a ribosome. Now, some ribosomes are free-floating in what we call the cytosol, which is the fluid of the cell. So cytosol is inside the cell. Matrix is usually the fluid outside the cell. I'm saying that one because that one took me a while, and I was like, they keep switching those words. Cytosol's inside. Yeah, anyway, so most ribosomes, however, are on this organelle, which is called the endoplasmic reticulum. The endoplasmic reticulum has a large surface area, and it has a bumpy part, which has all the ribosomes on it, and that bumpy part is going to do the protein synthesis. The smooth part, where there are no bumps, does toxin and lipid absorption, so it does detoxification, and that's its job. So those are the two jobs of the endoplasmic reticulum: rough ER and smooth ER. After you've made a protein, it'll go to the Golgi apparatus, which is what we see up here. The Golgi apparatus is kind of like a post office, where it will take in mostly completed proteins and other things, and complete them, and then it will send them where they need to go. So it'll refine them, and then it's either going to send them inside the cell or outside the cell. If it's going outside the cell, it will put them into a tiny little bubble, and then that bubble will go and touch the membrane of the cell. And since the bubble is made of the same stuff as the membrane of the whole cell, then when they touch each other, just like two soap bubbles touching each other, they will become one. And so if you've ever seen soap on top of water, when they touch each other, they can come together into one large bubble. That's what happens here. And so moves towards it, touches it, merges, and then ejects all the things that are inside. Moving to the next one. Next we have the mitochondria. Mitochondria are, this is the one that I think teachers like to say the most. You guys remember what the mitochondria are? The powerhouse of the cell, yes. Favorite phrase. All right, so mitochondria. Mitochondria have another function that isn't often talked about, but it's super important. And that is detoxification. And so we already found that smooth ER does detox. Mitochondria actually does that too. It has a lot of enzymes inside of it that are called, well, there's a variety of them, but some ones that are commonly talked about are CYP, CYP enzymes. So CYP P450 might ring a bell. If you've heard of any of them, it's probably that family. And there's a whole bunch of them, and you'll talk about them more later. And if you have genetic, this really helped when it clicked for me. So in later classes, you'll talk about CYP this and CYP that, and they're genetic mutations. And so when you start talking about genetic mutations, like in environmental medicine and things like that, those are a lot of the enzymes that are inside the mitochondria, so that you know where to place them. So if you have stuff going wrong with the mitochondria, you can have detoxification issues as well as energy issues. And so that's kind of what I take away for the mitochondria: energy and detox. All right. After that, let's see. Lysosome. Okay, so we have a lysosome. And the lysosome is going to degrade a lot of the proteins that when we don't need them anymore. And so the lysosome is kind of the garbage chute, if you will. It's the recycling station. Okay? And we're actually going to talk about a lot of diseases in the lysosome today, but basically it's if you can't get rid of things, then you're going to have issues, and there's structures that you'll build up, but you need to be able to break them back down, and if you can't, then that's a problem. There are some other things about the lysosome as well, but that's probably the major takeaway. Next.
Is that it degrades proteins?
Yes.
Okay.
So it's the garbage chute. Next, we have the peroxisome. Peroxisome. And I wrote that in black. We'll just say it's because it's the last one. All right, the peroxisome is going to, you'll notice that it has ox in it, per-ox. That's because it has oxygen radicals in it, and it can help with immunity and things like that. It also will degrade, I should say not degrade, but, well, degrade's okay. It cuts up long-chain fatty acids. Long-chain fatty acids that cannot be sent to the mitochondria. I didn't mention this because it's not what it's part of making energy. We can make energy out of fatty acids, so the mitochondria will do that, but it can't do the long-chain ones. It can only do medium and short. So long-chain fatty acids, like 26 carbons would definitely be a long. I think higher than 22 carbons. I don't think she'll ask you that, but really long chains have to go to the peroxisome. They can't go in the mitochondria. And so the peroxisome will break them down. It's not quite as energy efficient, but they take care of them. If your peroxisome's broken, you'll have a disease called Zellweger's, and we'll talk about that more, but basically Zellweger's. And you're going to have a lot of long-chain fatty acids in your bloodstream and everywhere because they have nowhere to go. All right. Last thing that I will mention, just because we have a disease about it, is the cytoskeleton. So I'm just going to draw a few lines down here to represent the cytoskeleton and kind of connect them. Essentially, it says the word skeleton in it, so that's kind of what you expect. It keeps the cell in its proper shape and gives it some resilience against mechanical injury. And so if you have that, like in red blood cells, for example, you have a cytoskeleton, and that's one of the things that keeps them in their normal donut, delicious-looking shape. And cytoskeleton. All right. So now that we've talked about those, and peroxisome wasn't the last one, cytoskeleton. All right. We have some diseases of the lysosome. All right, so lysosomal disease. Now, a lot of these are going to be because of a buildup of some kind of protein, because something in the lysosome isn't working to be able to break it down. A few of them are a little bit different than that, but that's kind of the theme, if you will. A lysosomal disease, one thing that I will say about all these that will help you on your test is they're all autosomal recessive diseases. All of the ones that we're talking about today, I should say, autosomal recessive, meaning that they are passed down with a recessive allele, and it's not through sex chromosomes. It's all the other ones. So that's these guys. All right. Lysosomal diseases. So I'm just going to, like, make sense in my head to kind of have 5 circles. And each one of them is going to be a disease. So we will start with inclusion cell disorder or disease. Icell disease is how it's typically talked about. So Icell disease. With Icell disease, you have inclusions that can't be handled, and the enzyme, and it causes mental retardation because of these inclusions that build up in the cells. Okay, and the key to remember that will help is the enzyme that's broken, and that is UDP N-alpha P. UDP NAP is how I think of it. And N is a really long word, and P is a really long word. But if you remember UDP NAP, then that's what's wrong with this one. It leads to mental retardation and at a very young age. All of these are with are early age when you see them. You can't go very long without being able to clear out proteins of a certain type without those building up and causing an issue. So all of these are young children. Okay? So for this one, I just think of someone who is napping. So they're napping. Okay. All right. Icell disease. And their pillow, incidentally, welcome to the world of In My Head, is in the shape of an eyeball. One of those stylized eyeballs. And so that's Icell disease and UDP NAP put together. Okay?
Oh, I get it now. I was wondering why the nap, but okay.
Yeah, exactly.
Got it.
All right. So next on our circle, we're going to go ahead and have up here, we're going to talk about Tay-Sachs. My favorite. Tay-Sachs, for a little while, they actually progress normally up to about the age of crawling-ish. It's really rough, like 2 years-ish. Don't necessarily write down the 2 years. That's kind of a roughly that time period. And they're neurologically and muscularly fine until then, but then they have enough buildup of the protein because of an enzyme called Hex-A not working. And so the particular protein that builds up is it's a type of glucosidase, I think. Don't write that down. It is a sidase of some kind, but Hex-A is what they ask you about is the enzyme. What enzyme is broken? So what happens when this buildup starts going is that you have neurological regression. And so they stop progressing and start regressing. And this can happen through a variety of ages depending on how bad the enzyme is. Is it completely gone or partially gone, kind of a thing. And it also, however, affects motor. And so they'll go backwards in motor as well. And they can have seizures and things like that. So this one I just kind of think of going up and going down. So this is just up and down. And on the way down, you add in seizures and things that were never there. Okay, and so who goes up and down? Tigger. All right. So this is my rendition of Tigger. All right. There's totally Tigger. All right. So,
You're so awesome.
yay. All right. Next, we have, oh yeah, and he has that's right. he has a magic wand because he's hexing someone. He's very excited about it. So he goes up and down because he's Tigger, and then he has magic wand that he's hexing people. So hex A. All right, next we have, that's probably why he looks so spastic. Next we have Gaucher's. All right. Gaucher's disease. Now, in Gaucher's, this one, I kind of, I'll talk about them separately. Yeah, we'll stick with Gaucher's. That'll make it easier. So Gaucher's is when you don't have the enzyme glucocerebrosidase. So gluco, like glucose, cerebro, like cerebrum, and sidase, which is sidase. Glucocerebrosidase. And so what that leads to, glucocerebrosidase. What that leads to is they actually get a protruding abdomen. It kind of looks, not trying to be funny, but it kind of looks like a beer belly sort of a thing, where it's a pot belly, I should say, perhaps, sticking out. And they have mental issues as well. And so okay. And to remember this one, I just have someone who looks like that because it's a very distinctive thing to see on a small child. And they also, in my mind, I just go ahead and stick a cerebrum on their stomach. So like that way I can remember glucocerebrosidase. And so they just have a lick and stick tattoo on their stomach, I guess. Okay. All right, let's go ahead and once you're done with your notes, go ahead and see if you can remember the 3 pictures or however you want to go through it, and then what were the details of each of the 3 diseases we've hit thus far. Hopefully the pictures in your mind are better than the one I drew. Okay, hopefully that was enough time. Moving forward. Next, we have Humps Disease. Or, since it has an E on the end, I often call it Pompe. It's Pumps, but Pompe's disease or Humps Disease. With this one, Gaucher's had a protrusion in the abdomen. This one kind of has a swelling in the face and the neck, and it actually makes so it's very hard for these people to breathe properly and respire, and it gets in the way. And they also have some muscle paralysis coming from this, and so this is happening kind of swelling towards the top of the body versus Gaucher's being a swelling in the abdomen of the body. And so I just kind of think of these as the 2 swelling diseases. And Pompe or Pumps and Pompe, I say Pompeii because I initially memorized it as, at the top, volcano, like Pompeii. And people after Pompeii had a hard time breathing. So, yeah, that's actually what we'll go ahead and stick right here. If you're familiar with the volcano Pompeii, it goes at the top. And then with all the ash in the air, people had a hard time breathing. And that's kind of what this one is.
You did say there was decrease in facial movements too? So that's
yeah,
neurological function?
It's motor function, yeah. Motor function of the face. And this is because of a buildup of glycogen.
Okay.
And so you can't actually break your glycogen down super well. And that ends up having the most severe issues in the face and the neck. And so it's breathing. And so when you see the volcano, think it's at the top. The volcano is named Pompeii, and the ash makes it hard to breathe. and eventually hard to move. All right.
Is there an enzyme that's associated with that? We're supposed to know, or no, we're good with that?
I believe it was like, it had the name glycogen in it, I seem to recall. I don't know at the moment, and I can't pull it up right now.
Okay.
So, this is the one where she talked to us more about the buildup than the enzyme.
Okay.
And so that's what I stuck in, and that
Perfect.
seemed to serve me well through the tests. But if you can find the enzyme, it'll be good.
Okay.
Just to have a full set.
Or maybe next week when we have the lecture, we'll know it. Yeah.
Right? Yeah. Okay, and then after this we have, let's see, Icell, Tay-Sachs, Goucher, Pompe, yes, Hurler. When you guys get to see the slide, this one has probably the most memorable picture. It's a child who is holding a baseball, and you can tell he's special needs, but he has a baseball and he's getting ready to throw it. It's like, oh, look, you can have a good life anyway, and it goes along with the name. Hurler, and so this one, the enzyme that is broken is iduronidase. And for this one, I just remember the kid with the baseball for Hurlers. And this also reminds me because the kid is a little bit, he's older than the kids you see in all the other pictures, and that's because these guys tend to last a little, they tend to do a little better. It's still not great, but I think the kid in the picture looked to be about 12, which is a better prognosis than a lot of these other diseases. And then the other thing that I did remember, iduronidase, if you'd like it, is, how many people here like Harry Potter?
Yeah.
Oh, good. We're among friends here. All right. So Ron, I'm not usually like a fan fiction or shipping person, but Ron is kneeling down to Hermione, and Hermione's going, I do, Ron. All right. I do, Ron. Iduronidase.
Oh, got it. Yeah.
So we have Ron kneeling, and this is the one that's probably stuck with me for like the longest out of all of these. And then Hermione's up here, and she's like, oh my gosh, I'm so happy. All right. So those are the 5 diseases of the lysosome in your tests. So these are the things that she's going to ask for, those enzymes. And in this one, glycogen, I really do think that it was the glycogen that she was more focused on for this one, for Pomps. She will also be asking you, is this a lysosomal disease or a peroxisomal disease? Which one of these is a mitochondrial disease? So we're going to talk about mitochondrial and peroxisome. But those are the questions I remember. More than, how old can they get before, like what's the median survival rate? She didn't go into that nearly as much, and that's why she skates over it during the lecture. Right now we're focusing on what happens if this part of the cell goes wrong. And so that's the kind of viewpoint we're looking from. All right, so if you guys would like to, go ahead and review the last 2 or all 5, up to you. All 5 probably would help make sure that they're up there. I'm going to go ahead and do the same since this one's a little bit back there for me. While you guys finish up, I'm going to clear the board. All right. Hopefully that went well. And we will now move forward with, I'm probably not going to label this up there because there's only one mitochondrial disease that you need to know. Woohoo!
Yeah.
So, if I can grab one of these, thank you. I'm actually going to go ahead and put him over here because if you remember, he's the one disease I wrote on the left-hand side of the board. That'll help spatial memory. So, down here, we have the disease Leber's optic neuropathy. And it's actually Leber, I think it's Leber-Kuhn's. All right, so Leber-Kuhn's optic neuropathy. So right in the name, we see optic. So this is going to be a disease of the eyes. Okay, basically, you have issues with chain gangliosides, retinal chain ganglions. That's what it is. Retinal chain ganglions. All right. So these are cells that are, well, they chain together, and ganglions are going to, it would take me too long to pull it back up. But basically, these are important in the eye, and if they break, you go blind. So Leber-Kuhn's optic neuropathy is a mitochondrial disease in which you go blind. And that'll, I remember that question. She's going to say which one is a mitochondrial. All right. So moving forward on that, that's all that you needed to know about that, is that having issues with mitochondrial, there are some diseases that are transmitted mitochondrially. That's the point of it, including that one, is you can have genetically inherited mitochondrial diseases. All right. And next, we have the peroxisome. And there's one peroxisomal disease, and that one's going to be Zellweger's. All right? So, this is the one that we're going to learn. And in Zellweger's, the peroxisome cannot accept fatty acids, long-chain fatty acids. The transporter is broken. I believe it was MCT1. Yeah, MCT1. Transporter is broken. I don't think you need to know the transporter. That was just added bit of knowledge. So, but the transporter is broken, and so the long-chain fatty acids, Fa for fatty acids, can't get in, cannot be broken down. And so they stick around, and they go everywhere. And so if you take someone who has this disease, Zellweger's, and take their blood, you're going to find a lot of long-chain fatty acids in the blood. They will have issues because of fatty acids, and that means that it will deposit. It can also lead to some calcium issues because fatty acids grab calcium. But if you were to measure just high fatty acids in the blood, then it's like, well, maybe you have a terrible diet. The way that we know that it's Zellweger's is comparing how many long chains do you have versus medium and short chains. Because if we can take care of medium and short chains, we'll have, like, you know, a little bit of these, a little bit of those, and a freaking ton of long chain in this. Medium and short chain are chewed up in the mitochondria for energy. Long chain don't fit in the mitochondria transporters, and so they go to the peroxisome. So this is our peroxisome. Peroxisome. And you end up having all the issues you would have with really high free fatty acids in the blood. There might be some other issues, but this is the predominant thing that she's focusing on, and that's what I'll go ahead and put forth to you. And I think this comparison thing right here with the long, medium, and short, I think that was something that was in a clinical. Remember that. It's important. Put it that way. Try and be a little less blatant. All right. Let's see. Yeah. So, if you guys want to take a moment to remember What is our one mitochondrial disease? And the really important thing is remember that that's mitochondrial. Separate it from it being other things. It's also important what it does, but make sure you don't forget where these are. And then what's our peroxisomal disease? Oh. I just realized in my head that I never gave you a memory thing over here. Basically, I think of that I can't draw. I think of a chain being swung around by a gang member. So he's got his handkerchief thing tied on top, and he's swinging around a chain. So chain gangs. So chain ganglions. And that's been helpful. And they're retinal chain ganglions. So he's swinging around a chain. Over here for the peroxisome, honestly, I didn't have a picture. So I just remembered it chewed up long things because she actually will have she'll talk about that one long enough. And it's supposed to be it says magnetic. I'm just going to show you that.
For the Zellwegers, is it what enzyme is not functioning?
So it's a transporter.
Oh, a transporter.
So it has all the right stuff inside the peroxisome, presumably, but it can't let the long chains in. Okay. So they never get in there and the job never gets done. So transporter. I'm glad you asked that. That's a good thing to remember. And MCT1 is the transporter. It also might affect MCT2. That's not going to be on the test. I can say that one pretty firmly. I think I even picked that up from a different class. But for the sake of completeness, because it's like, what transporter? All right. That's Zellwegers. All right. And moving to our last disease of the specific pieces. All right. I'm going to go ahead and get rid of Zellwegers. I should have just kept lysosomal. Anyway. I'm going to go ahead and put cytoskeleton down here. So that's what a cytoskeleton looks like, and it's holding the shape of a cell. Okay? And so this cell is a red blood cell. Okay? And they're supposed to look like donuts, which is why donuts are good for you. The more donuts you eat, the more red blood cells you have. That's why America has problems. Is that a fact? Don't quote me on that one.
Okay.
All right, so red blood cells, they get their shape from the cytoskeleton. Okay? Just like all others. So, skeleton. All right. Whatever. Spelling's overrated. And if the cytoskeleton is broken, what do you think the native shape of a cell would be? A sack of water.
Essentially.
A sack of water, yes. It would be just a sphere. And so if your cytoskeleton has issues, then you're going to have a sphere-shaped cell. And so they call this disease hereditary, because it's passed on, like all of them that we've talked about, spherocytosis. So sphere, meaning sphere, cytosis, meaning cyte is for the cell, osis, meaning condition of, hereditary spherocytosis. All right? So this is, hereditary spherocytosis. And the molecule that has the most issues that she wants you to remember is a molecule called spectrin. So spectrin is part of the cytoskeleton. And if spectrin isn't working properly, is not able to be shaped properly, then the cytoskeleton cannot function. I'm pretty sure it's part of the cytoskeleton. It might be a support protein. But spectrin, then, if that's not working, you get hereditary spherocytosis. Your red blood cells turn circle, and that means when they're going through really narrow capillaries, or when when they go to a junction where the blood cells diverge into a Y, and it's like, are you going to go left or are you going to go right? They can hit the walls a lot more because they're not as aerodynamic, but for water. And so they'll hit walls a lot more, and they're also a lot weaker. And so both of these things lead to them breaking a lot more and a lot easier. And so you can end up having anemia, and you can end up having blocked vessels a little bit. And so basically...
So it's not produced, spectrin?
Right. Spectrin is not produced, and the reason, is because it's passed down. It's a genetic mutation. And hereditary spherocytosis, you'll eventually learn a group of diseases called, they're diseases that break red blood cells, and this is one of them. And so they're hemolytic anemias. So why do you have anemia? Because your red blood cells are being broken. So hemolytic, heme for blood, lytic for breaking, anemia, you have low blood oxygen, low red blood cell count. So, yeah, that's that disease. All right. So from there, let's see, that's all the diseases. After that, we are going to talk about surface receptors and everything. But before we do that, any questions? Okay. Go ahead and review just the last disease if you want. Maybe the last two, since I erased one up there. And I'm going to do that really fast, and then we'll move forward without too much of a gap here, because I want to make sure that we get through the receptors pretty well. But I also want to make sure that this is more than hearing it and dropping it. Okay. Yeah, all good. Okay, so, one thing that I'll throw into the hereditary spherocytosis is just a picture, because I read the pictures in my head and then I forget to draw them. It's a friendly little ghost. He looks like a spectrin. A word for ghost is specter. So if you want to stick a ghost there to remember, that's helped me and some of my classmates. All right, moving forward. He's like in the gumball. It's a ghost gumball going through your arteries. All right. And now, right, receptors. So this is a cell. And we have three major families of receptors. We have ion channel receptors, and those let little ions go through, and we're actually not going to talk about these too much. They exist, they are a family, they can let sodium and potassium and all that stuff go through, one way or the other, and just know they exist. You've talked about them with Professor Schmidt, I believe, and so, yeah, that's a superfamily. Another superfamily of receptors are the third one, yeah, I want to say them in order though. G proteins. So the G protein coupled receptors. These guys have 7 proteins all put together to make so that they can go through the whole membrane, and you have something bind on the outside. I'm drawing 7 circles, and then a big G protein circle at the bottom. And up here you've got like the receptor bit, which is part of the protein. I'm making it stick out a little bit more. So you have a bunch of proteins all grouped together. There's a G on the inside and a receptor on the outside. You have something bind right here, and then that sends it through all this sequence of proteins, and then the G protein is going to go and either go through the adenylate cyclase pathway, which we will talk about in depth, or the phospholipase pathway. Phospholipase C. Those are the two pathways, and there's a total of 5 different categories of G protein receptors, and they're called things like beta 2 receptor, alpha 1 receptor, alpha 1, 2, 3, beta 1 and 2, I think are the 5. You aren't expected to know all of those at the moment, but I'm just mentioning them. So they will go through, and yeah, some of adenylate cyclase and phospholipase pathways. Moving on to the third one, those are the kinases. Kinase receptors. So, question for you guys. It's my favorite enzyme. What do kinases do?
They put phosphates on things.
Yes, they put phosphates on things. They put them on, or if they go in reverse, they take them off. And that's exactly what happens with these receptors. And so you have, it's another protein that goes through the membrane. It's not a G protein though, but it is one that goes through the membrane. And on the end of it, it has a kinase, or it talks to a protein that has a kinase. And so sometimes it's on the receptor itself, and sometimes it's on something that kind of sits around next to it until it gets kicked by the receptor. Okay? All right. So, and those are the kinases. So G proteins, I'm going to go back to G proteins and tell you a few details about each of the pathways. Adenylate and phospholipase C. And I'm going to move the kinases like to the bottom so we'll have room later. All right, so G proteins, they have, in the adenylate cyclase pathway, they are going to have the G protein go and turn on adenylate cyclase. Adenylate cyclase will take AMP and it will bend it into a circle. And then we call it cyclic AMP. And then that is going to go and do a bunch of stuff inside the cell, because it's a secondary messenger and it will turn on whatever it needs to turn on. Make sense? Mm-hmm. Okay. I'm going to add one more thing actually. Cyclic AMP typically goes to protein kinase A. And then we'll stop there. And protein kinase A goes ahead and does a bunch of kinasing for the same purpose. The reason I wanted to add this one more is because you'll notice that adenylate cyclase starts with A, protein kinase A. Those two always go together, and that could be a question at some point. And if you always know protein kinase A is over there, and adenylate cyclase is over there, and they go together, then that might help. Phospholipase C is actually in the membrane, so we'll go ahead and move the arrow to the phospholipase C, and then we'll say that's what that thing's called. Okay? That is phospholipase C. Phospholipase C will take PIP2 and break it, because it's a lipase. It breaks lipids into pieces. And so PIP2, or phosphoinositol diphosphate, is just part of the membrane. It's always there. So phospholipase C is literally just drifting through the membrane until something pokes it, and then it'll reach out a hand, grab a thing, and break it into, and let it go. The two things that come out of PIP2 are going to be IP3, which is going to go and talk to the smooth muscle, or it's going to go and let calcium out. This usually will happen in a smooth muscle cell, this whole pathway, to make so the smooth muscle cell will contract. But it's going to let calcium out regardless of what kind of cell we're in. Calcium from the smooth muscle, or the smooth endoplasmic reticulum, SER. the other thing is going to be diacylglycerol. And diacylglycerol, which goes and talks to protein kinase C, PKC. And then that'll go ahead and kinase a whole bunch of stuff. Similar to protein kinase A, except this one's protein kinase C and stays in the membrane. Those are the two pathways. And that's all I'm going to say about them. Any questions? Did the sequence make sense and everything? Okay. All right. Got in a bit of physio review, and I don't need to erase that. I'm eraser happy today. All right, and for the kinases, we have 3 kinase families. Maybe I do need to erase a little bit. Get out of my way. All right. Kinase families. I'm going to draw another cell. We have tyrosine kinase, tyrosine, that is a tie. Alright, tyrosine kinase has its own kinase. That's kind of what makes it cool. You actually find this guy, and this is something that she'll mention, you find this guy in the pancreas. And so when we were like, hey, we need insulin, and get those beta cells cranking it out, you got a tyrosine kinase receptor. And it'll come down, and it'll get poked, and then it'll go ahead and on this side, on the inside, when it is poked, it will go ahead and use its kinase activity to phosphorylate, like all kinases do, but it phosphorylates itself, which is really strange. And once it phosphorylates itself, it activates a different part of it that goes ahead and does all the important things inside the cell. It starts the cascade. We call this autophosphorylation, when you go ahead and stick phosphate on yourself. Auto, same, autophosphorylation. Okay? So, this was the tyrosine kinase family.
You said tyrosine kinase gets poked and then it phosphorylates itself in others?
It phosphorylates itself. I believe it phosphorylates others. I don't recall saying that. I believe that's true, but it definitely phosphorylates itself, and then that other part of it will go ahead and I believe it acts through phosphorylating other things after that. But what you want to remember for sure is this is one that's in the pancreas, beta cells, and it autophosphorylates and then does the cascade with the last part that just got activated by phosphorylating itself. So you can kind of make this into 2 different pieces, where there's the first part that's the kinase that'll go ahead and do the phosphorylation, and then the part that gets phosphorylated will do other things. But they're technically all connected. All right, number 2. So that was number 1. Number 2 is the JAK-STAT pathway. JAK-STAT. Okay, so with a hyphen in between them. So the reason they're named this is because you will do things with some JAK proteins, and then you'll do something with a STAT protein. So there are 2 JAK proteins and 1 STAT protein. So first you'll hit JAK number 2, then you'll hit number 1, and then you'll hit STAT. So the receptor is on number 2 turns on number 1 turns on STAT, and then STAT will go to the DNA and do whatever it needs to do. And so this one goes to genetic material, DNA. So I just think of like 3, 2, 1, except it's 2, 1, STAT.
Wait, so what hits the 2?
These are receptors, so whatever was in the blood that's going to turn it on. So some kind of hormone. And this will go and turn on DNA, and it will let out a whole bunch of factors that do the job that's needed. Third receptor family, third kinase receptor type is going to be the SMAD. All right. And I just realized that I jumped ahead of myself. What I told you was correct, except the 2, 1, and then SMAD. Over here, it's actually just JAK directly to STAT. Double check this part, because now that I've said it one way and another, I want to be careful, but I do know that the third one is SMAD, and it goes down to genetic material as well. And you'll notice that the only one out of all three of these that has autophosphorylation is the tyrosine kinase. So go ahead and double check me on the end there, and that's everything for the lecture. Any questions?
Thank you.
Yeah, no worries. All right, so the point of all of this was what's going on inside the cell and what happens when you break what's going on inside the cell. Those are the diseases and all the organelles. And then after that, receptors. How do we talk to the cell from the outside? So hormones are coming. How do they talk to it? And what can they bind to? And these were the receptor families, ion, G protein, and then our various kinds of kinase receptors. All right. Thanks, and that's it.